In this video, I will go over the Affymetrix GeneChip, which is the most common DNA microarray platform. This chip, which can fit in the palm of a human hand, can be used to detect expression of thousands of genes. Let's first discuss what I mean by the term gene expression and why this is important. The central dogma of biology is that DNA is transcribed to RNA, and then the RNA is translated to proteins. Reverse transcription, which is relevant to this video, is when RNA is converted to DNA, a process that happens routinely in many RNA viruses. Genomic DNA contains all the genes in a genome, but doesn't actually tell us if those genes are actively being transcribed and used in the cell or not. A good analogy is that DNA is like a cookbook containing thousands of recipes, and the RNA is a copy of just certain recipes that you want to make. So we have to look at the RNA to see which genes are more expressed than others. And why should we care about this? By analyzing 2 or more samples, you can compare how gene expression is different between them. For example, how a cancerous mouse sample expresses differently from a non-cancerous mouse sample, or how a plant sample expresses differently under drought versus non-drought conditions. Now let's get into the specifics of how an Affymetrix GeneChip works. The GeneChip itself is a plastic cartridge containing a glass slide that has many short pieces of DNA, aka oligonucleotides or oligos for short, attached to it, which represent most of the genes in a genome. The total RNA is extracted from your sample of interest, converted to complementary DNA by reverse transcription, and then transcribed to complementary RNA in the presence of biotin-labeled nucleotides. Biotin is a non-protein molecule that binds strongly to specific proteins like streptavidin, and in this process, it's used to tag the cRNA to be detected. This cRNA is then fragmented into smaller pieces using chemical hydrolysis at high temperature. The resulting cRNA probe is then added to the gene chip and incubated to allow the cRNA to bind to the DNA oligos on the chip. Next, wash off any cRNA fragments that didn't bind, then stain with streptavidin linked to phycoerythrin. The streptavidin is a protein that will bind tightly with biotin, and the phycoerythrin is a pigmented protein originally derived from red algae that emits light, i.e. fluoresces. Finally, scan the chip to detect fluorescent spots and analyze the data using the GeneChip software. The genes that are expressed in the sample will produce bright red spots on the chip, and those that are not expressed will stay dark. The brighter the spot, the greater the respective gene expression, so quantitative information can be obtained. Several thousand genes can be examined in parallel. Now let's dive into understanding how the oligos are designed. 2 25 nucleotide oligos per gene are used. 1 oligo matches the gene perfectly, and 1 contains a deliberate mismatch in nucleotide 13. The mismatch oligo serves as a control to account for nonspecific binding. During the data analysis step, the expression from the mismatch control oligo is subtracted from the expression of the oligo with a perfect match to remove any noise coming from nonspecific binding of the oligo. Once a microarray chip is designed, many identical copies of the chip can be produced and run in parallel, which is a significant advantage of microarray technology.